Hello, my name is Suman Rice, and I'm chair of the Society for Reproduction and Fertility. I'm delighted to welcome you to the SRF 75th Anniversary Lecture Series. This collection of talks celebrates 75 years of the Society, its rich history, and its ongoing contributions to reproductive science. The Society was founded in 1950 as the Society for the Study of Fertility, bringing together a remarkable mix of scientists and clinicians, from reproductive physiologists to endocrinologists, pioneers of livestock breeding, and experts in human fertility. Each lecture in this series highlights the diversity of research and innovation in our field, reflecting the interdisciplinary spirit that has defined the Society from the very beginning. We hope these talks give you a sense of the Society's achievements, the work of its members, and the dynamic community that continues to advance reproductive science.
I'd like to begin by thanking the Society for Reproduction and Fertility for inviting me to give this presentation in remembrance of the 75th anniversary of the SRF. It's quite a privilege, and I appreciate it very much. Now, I'll be talking about the place of reproductive sciences in wildlife conservation. That's the topic that I've worked on for many years. It is a fairly complicated topic, and I'd love to have a chance to give you a snapshot of what I have to say. But just to begin with, there are a few things that I wanted to point out, not necessarily in detail later. The slide, the picture at the top of this screen on the left-hand side shows some coral, and the reason for putting that there is you might not think it's necessarily reproductive biology, but these days with coral bleaching being a major problem, there's a lot of interest in coral cryopreservation, or indeed coral preservation, and then restoration as well. And this has seen some real advances in cryopreservation technology to the extent that that coral is now being used to reconstitute some of even the Great Barrier Reef, where there are major problems. On the other side of this slide, I've got a couple of pictures of a species called the black-footed ferret. Now, I won't be talking in detail about this, but the reason for mentioning those is that this is one example where a species went almost totally extinct in the 1980s. There were only 18 animals left at one point. And because of reproductive biology and technology, in fact, like artificial insemination and so forth, that that species has been restored to the point where it has been possible to reintroduce them into America, where they belong. And the population is now very healthy and actually in the region of several thousand. Now, at the bottom of this slide, I just wanted to mention here we have a gazelle from Saudi Arabia, and I want to mention a little bit about that later. I'll mention a little bit about the Asian elephant, which is basically an endangered species at the moment, and people are trying to use technologies to breed those animals. And at the bottom on the right-hand side, I've put a picture of one of the frogs and toads. The reason being that there is a huge problem with some basically poisonous fungi that are devastating frog and toad populations across the world. And I wanted to make a mention of those. Now, people might think that conservation biology in relation to reproduction is just about saving species and doing or simple technologies to breed animals. But I just want to make the point that it is concerned with more than that. It's concerned with the maintenance, loss, and restoration of biodiversity. So it is actually a very wide topic. Now, in terms of what you can do with conservation and how that reproduction could fit into that scenario, I just made this simple slide to say that there are various levels of complexity, and whilst habitat management and protection might seem to be the easiest option, it is actually really difficult because it involves a lot of landscape and um, interference with landscapes. Now, nevertheless, that can be appropriate, especially if things like translocation of animals from one place to another, perhaps to a better environment, is contemplated. So that could be done by just simply moving animals from one place to another, or in the case of a species which might have disappeared totally from an area, to actually reintroduce those species. So quite a lot of that is going on even in the UK. Those are probably not things that you would regard as technological, but technology is now used quite a lot, but in specific reasons, for specific reasons. And I've just put here artificial insemination can be used quite a bit. Semen freezing and artificial breeding, assisted breeding, can be used quite successfully. And then there are other technologies which I won't go into in detail, but just to mention cloning and bringing back species that have gone extinct. So I won't really mention those again, but I just wanted to, for the sake of completion. Now, you might think that technologies have to be really complicated. And what you really don't want is to have technologies that are so complicated they upset the animals that you're really interested in. So a good starting point would be look at technologies where you don't actually have to do anything invasive to animals at all. And this is an example of this, being able to measure reproductive hormones from substances like either urine or feces. Feces are especially useful. And the sort of thing that can be done is to, in this example, this is a Saudi Arabian gazelle, where people were interested in understanding whether those animals are undergoing reproductive cycles or not. It was possible to collect fecal samples on a daily basis. Because these are desert animals, these fecal samples are pretty dry, and they are relatively straightforward to measure progesterones in those. And you can see here that by doing that measurement, it's possible to identify um, individual cycles, reproductive cycles, and even the luteal phases and the follicular phase. That is particularly useful, and it doesn't interfere with the animals at all. If, on the other hand, you want to know whether animals are pregnant, and this is another gazelle species called a Mahmoud gazelle in a captive population in the south of Spain, but originally from North Africa. People might do artificial insemination, and then they want to know how it works. And it is exactly a project that I was involved with, using the same principle and measuring progesterone in those daily collected fecal samples. It was possible to establish that a particular female was indeed pregnant. So this is a very good non-invasive pregnancy detection test. Unfortunately, the sort of thing that might happen and did happen in this case was that the people doing the work were very keen to know if the insemination had been successful. And so they went for ultrasound measurements, which unfortunately caused so much stress that although the AI was successful, the stress caused resorption. So at least we know that's the way not to go. But in principle, the non-invasive pregnancy detection is a very useful way forward. And that this principle can be extended further. And this is a very nice paper where people in Canada used fecal sample analysis for the endocrine side of things, but also to analyze the DNA, and they analyzed the physical attributes of fecal pellets. So using that approach, they were able to not only determine whether they have males or females present, which is something otherwise you would need to go out into the countryside or into the bush, whatever it is, with binoculars and actually look at the animals. But, and using this approach, you can take these different samples and basically without leaving the lab and do lots and lots of finding out about the population. And really, this is what people want to know: is the population in a good state or not? And the only other way of doing that at the moment is just to go out with a Land Rover and a pair of binoculars and really look for the animals, try to figure out what's going on. So this is a very useful approach. Now I'll swap slightly, change tack to figure out, to mention things which I've actually been a little bit involved with. I was involved with a project to try to develop more knowledge of reproduction and reproductive in the males of Asian elephants in Thailand. There's a big, a lot of um, a lot of elephants in Thailand, which used to be from um, logging centres. Now those are basically kept as exhibits, really. But nevertheless, it's important to develop things like knowledge of reproductive physiology and understand about inbreeding and try to avoid inbreeding problems and so on. So scientists in Thailand are really trying to develop techniques, perhaps even for artificial insemination. The good thing is that they have nailed the technique for semen collection in these male elephants, so it isn't terribly invasive. But the bad side of that is that the semen in these Asian elephants is actually of really terrible quality. And so there's a lot of need to investigate why that should be and whether it's possible to understand it and even improve it to the point where it could be used for artificial insemination. We also understood now that the sperm from these Asian elephants is very, very delicate and easily damaged by procedures like cryopreservation. I mentioned the importance of the frogs and the toads and the breeding programs, and the reason for this is that there is this fungal disease that is going around the world, devastating populations, and some of these populations are now just on the verge of extinction. This picture is of a particular species, Puerto Rican crested toad, where it's a really high priority to try to develop artificial breeding techniques. And so actually the use of these techniques with frogs and toads is now becoming perhaps even more successful than with other species. And I could hear a slide. I won't go into this, but it's a slide from a colleague in the University of Newcastle in Australia, where they are really keen on making developments for breeding amphibian and various types, a lot of endangered amphibians in Australia, and they're developing cryoconservation techniques and able to recover their sperm and use them for breeding and supporting the populations. Now, I wanted also to mention how the understanding of reproductive physiology is really important in a case such as the giant panda, which people have been keen to breed in captivity for many years. And it's only recently that through a massive multi-center approach that it's now understood how to breed those animals in captivity. And it was due to some actually very fine scientific technical work that became clear how to use precision timing to make sure that those giant pandas bred. And the reason for putting this graph here is to show that with pretty intensive collections of urine, it was possible to identify, for example, an estrogen peak. Here we have an estrogen peak showing up uh, just before ovulation, and LH peak also being timed together with the ovulation. And it was known, calculated from the information that they had, that the greatest success for artificial insemination, it was achieved when the insemination occurred within 40 hours of the decline in estrogen. So you see, knowing this level of detail is actually really, really difficult unless you have that precise information. Now, I just wanted to mention also that I've just touched on some of these topics, and that there are many other programs going on throughout the world using these techniques and other techniques as well. I've just mentioned here the application of such techniques with marine mammals and with fishes. I mentioned the coral reefs at the beginning. It's now known that parental diets have a major impact on the health and survival of offspring, so that sort of thing is being studied. We also know now that endocrine disrupting chemicals can be a major problem, again, interfering with conservation. And on the other side of that, contraception can be a useful management tool to control populations. Myself and my colleagues, Pierre Comet-Soley and Janine Brown, have published quite a lot of papers about all this, and we've put this all together in a book, Reproductive Sciences in Animal Conservation. This is the second edition, and the third edition is almost ready and is going to be published in early 2026. So just to finish, I just want to emphasize that there's a lot that can be done with reproductive biology in the context of biodiversity conservation, but to emphasize that it is extremely complex, and each time you try to work with a particular species, you probably need a specific project. At the moment, it's not the sort of thing that can be just done in 5 minutes. So just to finish by saying, I'd like to acknowledge the help that I've had over the years from the various colleagues across the world. I've even worked on seahorses and marsupials and so on as well. And really to acknowledge that none of this can be done in isolation. And so I just appreciate all the help that I've received.